After performing these experiments and after we have finished all these topics, the next topic to move on to is the dihybrid cross. Now Mendel gave his like whatever we have learned so far, that means the unit that the factors are in pairs and one is dominant, one is recessive, or otherwise you can say law of dominance and law of segregation. All these things you have learned so far are based on the monohybrid cross. Now Mendel performed dihybrid cross and he came up with another law, and that is the principle of independent assortment. Now what is the principle of independent assortment? So by this cross it is going to become clear to you. A dihybrid cross is performed to understand or to study the inheritance or movement of the alleles of genes or factors which control two different characters. So in this topic what you are going to understand is that two character controlling genes are taken simultaneously. Now the parental generation, remember, he performed such combinations with many other, because 7 pairs of characters he had, so he had many combinations to choose from, and he performed dihybrid also. Remember, he performed trihybrid crosses also. So he chose 2 parent plant, for example, this is NCERT example only. So round and yellow seeds, and then wrinkled and green, because round is dominant over wrinkled and yellow seed color is dominant over the green seed color. So in the parental generation he took the seeds. Now those seeds were again made pure line. Please remember, pure lines means capital R, capital R, capital Y, and here the genotype of the parental generation is small r and small y. Now because this individual is a pure line and pure line, how many types of alleles are there? Just one type, R. So one allele of a gene is R and the other allele of the factor, better to say, is Y. So in the gamete form there will be only 2 alleles possible, one from R and other from the Y. So the gamete will carry which genotype? Because gametes are pure. Pure means they carry only 1 out of 2. 1 out of 2. So it will carry R and Y. And in the same manner, this plant will prepare the gametes, and those gametes will carry small r and small y combination. Please remember, don't show capital R in a gamete, or don't show small y in the gamete. This is wrong, because out of 2 only 1 will enter into the gamete. So now what will happen? These gametes will undergo random fertilization, and because all gametes of this individual are same and all gametes of this individual are same, only one result is possible in the F1 generation, and that is capital R, small r. Always remember, write same alphabets together. Don't write R, Y. You have to mention R, capital, small r, capital Y, small y. Capital should be written in front and small should be written next. So capital R, small r, capital Y, small y is the F1 genotype. Now because capital R is completely dominant over small r and capital Y is completely dominant over the small y, so the phenotypic expression in the F1 generation would be all are heterozygotes but all are dominant in feature, that because round is able to express, R will not express, Y is able to express, small y is not able to express. So round and yellow will be the phenotype of the F1 generation. And when selfing is done, now I will tell you today how do you do calculations to find out the types of gametes, etc. So when the selfing would be done, now in selfing, remember you will be making every combination. R will go with capital Y also, R will go with the small y also, small r will go with capital Y and small r will go with small y. So you can see same things I have mentioned here. Because it is selfing, so from the pollen grain, same information. Selfing means no two genetic makeup, similar makeup. So the information from the pollen grains and information on the stigma, or you can say in the female portion, egg. So male gamete and female gamete and male gametes are going to be with similar information. So capital R, capital Y, capital R, small r, capital Y and small r, small y. These are the genotypes of the 4 gametes possible. So 4 gametes from female side, 4 gametes from the male side, types I mean to say, not the number, types. So when they undergo random fusion, these are the combinations possible. And in the F2 generation, the phenotypic ratio would be round and yellow, wrinkled and green, and now to your surprise, combinations, new combinations are coming. So round and yellow, wrinkled and green are just like the parental generation, but the round and green and wrinkled and yellow is the recombinant. Remember recombinant topic of your 11th class, that zygonema, then zygotine, then pachytine, then diplotin, that same concept, recombinase enzyme. So crossing over is going on, because of which round will go with green and wrinkled will go with yellow also. So opposite combinations are also there. And the surprising fact here is that the phenotypic ratio is going to be 9:3:3:1. So this is nothing but an easy calculation of 3:1, because it was 3:1 in the monohybrid cross. So 3:1 raised to the power 2. It is going to be 9:3:3:1. And genotypic ratio over there was 1:2:1. So here it is going to become 1:2:1. Then 2:4:2, then 1:2:1. Anyway, you can write, it's easy to remember. 1:2:1, then 2:4:2, and finally again 1:2:1. So how do you do these calculations? We are going to learn today. And before that, just remember one thing. By this cross, Mendel gave another conclusion, and that is today considered as the principle, but he only stated that when two pairs of traits are combined in a hybrid. Now how many are there? When two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other character. Now you can see here, capital R is for round trait, small r is for wrinkled trait, capital Y is for yellow color trait, and the small y is for the green seed color trait. So how many traits are there? 4 traits. That's why NCERT, the note is telling you that two pairs of traits when are combined in a hybrid, this is a hybrid, segregation of one pair, where one pair means RR, is independent of the movement of the other pair, Y and Y. So movement means how are they going to pass into next generation. So the movement of the capital R and small r is independent of the movement of the capital Y and small y. This is the principle today is called as the principle of independent assortment.